So, Zach, I really appreciate you taking the time. What have you been up to lately?
Yeah, mostly just been up to keeping Zbiotics moving forward and, trying to figure everything out and really trying to adapt on the fly as the world is just constantly changing, right? Like we launched in a pre-COVID world in the before times and then, COVID happened and obviously everybody changed like all their social behaviors and
Hmm.
then now it's definitely not, we're still in that time, but as it looks like people are, starting to, there seems to be a light at the end of the tunnel here with the vaccine and people's behaviors might be sort of shifting back to what we knew before. And so just kind of thinking about how we kind of move with all
Hmm.
those things. So that's kind of
Yeah.
what, on these
Nice.
days, pretty much.
Nice, man.
Yeah.
Yeah, that's awesome. Yeah, so everybody I think that's watching this right now has just seen the original TikTok video. You have a product now that can kind of cure hangovers. This is like a huge thing for anybody that's in university, anybody that's still drinking. And the main thing is that it processes the acetaldehyde in our gut. Can you explain kind of how you came to this product and how you built it?
Yeah, totally. I mean, I think that like one of the amazing things I discovered so a little bit of background, I did a PhD in microbiology. And while I was in my PhD, we use genetic engineering all the time to make research tools and just sort of like study bacteria and stuff. And I just found that it was like we take this thing for granted, right? Like this technology so much, it was just so easy to do these things. And we're basically just leveraging a process that bacteria have refined over the last 3 billion years. We think of like, when we think about, for instance, to digress ever so slightly, think
For it.
about like, plant crossbreeding, right? Crossbreeding plants, right? And it seems like a very natural and simple thing that we do to kind of essentially exchange traits between plants. And we think of that as this like very basic and simple and old thing, right? Like, but it's actually like really rudimentary and not at all well refined relative to bacterial gene transfer, because bacteria have been around so much longer than plants, several billion years longer than plants. And
And their life
so
cycles are faster. So then
much
they evolve
faster.
faster. Yeah.
Exactly. And they're much better at exchanging DNA, incorporating DNA. They're much more precise. They've had a lot longer to essentially get it right. Right. And so when we look at like the way bacteria exchange genes, it's so much more efficient than plants. And so we can be so precise and so clear about what we're doing. Whereas when we cross two plants, we have like no idea essentially what's happening. We kind of just like this black box, and we hope that it has the things we want and not other things. But that's not at all the case when we're editing bacteria. And so in any event, and this is something that is just like, fundamental and well understood kind of in the world of genetics, like especially microbial genetics. And so I was working on this stuff and it just struck me like how cool it is that we can, with genetic engineering, we can program any biological function on the planet, that anything
Mhm.
in the whole planet does. We can program that into a bacteria using genetic engineering. And I just think that is just amazing, right? And so,
What were you doing in your PhD? What were you studying?
well, it was something very esoteric and like having
Of
no
course.
impact in the real world, but it was just like an interesting question, as is the case a lot of times with academia. I was studying this bacteria called Legionella pneumophila, which causes a rare form of pneumonia called Legionnaire's disease. And, I got just interested in this interesting kind of way that the bacteria was regulating its genome. And I was just studying this kind of random family of genes that were very, very interesting academically speaking.
What do you mean, not to dive too much into this,
Yeah,
but like, what do you
yeah.
mean by regulating its genome?
Well, so like this is one of the cool things about bacteria is that they have this, like we think of them as these sort of like rudimentary beings, like they're single cells, they're very simple, and they are in some ways, but they're actually, in some ways, it's actually a lot more complicated, right? That like they have to in one cell accomplish like all of their life functions that like we use a lot of cells, we sort of specialize and outsource, right? Like a skin cell and a heart cell do kind of two different things for us. But
Mhm.
like with the bacteria, they kind of got to do everything. And so that means that they have to make a lot of decisions like on the fly in a given environment in order to be successful in that environment. So, if there are nutrients available, you don't want to spend energy swimming around. You want that system off and then turn
Mm.
on like nutrient acquisition. Or if like you're under attack, you want to shut down everything else and turn on like, defense mechanisms or
So,
counterattack. And so,
so is it essentially like regulating its epigenetics
yeah,
on the
it is.
fly?
It's like that. It's like, what genes are, they have this cohort of, with Legionella, I think it's something like 3,000 genes, and they, want to express some sometimes and others other times. And that has got to be very situationally dependent. And so I thought it was a really fascinating question, like how so for instance, with Legionella, when it's growing exponentially, it turns off all of its virulence traits and things like that, because it's really focused on just replicating. And that's usually in a situation where nutrients are plentiful. And then when nutrients become depleted, it like really quickly shuts everything off for growth, and then it switches on all these like these defense and virulence trait, factors and things like that. And it's sort of like, how does it do this whole like global shift? And so the bacteria, what the I was studying was like this really fascinating and interesting mechanism, because all different bacteria do it different ways. And they had this sort of like master regulator called CSRA, and it was part of, I mean, there's a lot of different things, but it was part of what helped kind of turn off certain traits and turn on others.
Okay.
And it was just really cool to learn about, and it functioned in this very, very, specific way. And then kind of, I found all these other genes that sort of looked like it, and then they were also conserved. And so it was like, okay, what are they all doing? And so that was,
so it had
question.
like kind of hidden genes that it wasn't necessarily using all the time. And you were like, okay, what do these do? Can I put it in an environment that will use these genes and then figure out what they are actually meant for? Like 3,000 genes, trying to map like what the genes.
Right. So these guys like had the smell of something important because they were related to these other genes we knew were very important. But as you pointed out, I wouldn't necessarily call them hidden. They were just sort of like hidden to us because we, grow bacteria in these luxuriant laboratory conditions. And so they don't use a lot of their genome because they don't have to. And so yeah, like you say, trying to like figure out what conditions kind of these things turn on.
So did you find any cool conditions, or what were you going to say there?
Yeah, no, actually it was. So Legionella is this really interesting bacteria that has this, like, it can persist in a lot of different unique environments, and sometimes very stressful ones by bacterial standards. And so we did find that one of these genes looked like it was important for Legionella's ability to persist in new sort of like more stringent environments. And so that could have ramifications given the fact that it is a pathogen. So that, truthfully, though, it was mostly academic. It was very fun to kind of be an investigator and kind of dig into all that stuff. But I think one of the big things I took away from that was that there were all these strategies that bacteria use to turn on and off genes. And I got, there were some really elegant kind of like counter switches that bacteria were using to regulate really important functions like motility, right? Which is a very expensive function. So the bacteria's ability to swim is super important for its survival, but it's also very expensive. Like it takes a lot of proteins and a lot of energy. So they want to be very careful about when they turn that on and off, but it's a very robust function. And so it like ultimately at the core of Zbiotics, right? We talk about this product that, breaks down acetaldehyde and all that kind of stuff. But like what actually our technology is at the end of the day is actually that gene regulation and the ability of us to edit the bacteria so that it will express the genes we want when we want them to, and not like when we don't, or when the bacteria are in an environment where they wouldn't normally express.
So were you able to make the motility genes so that it could swim around express when it necessarily didn't want them to based on the environment? So then you could be like, okay, it's in this environment, it wouldn't naturally be swimming, but we're actually going to make it start swimming.
Essentially, like we could, right? Like, and with Zbiotics, I actually did end up leveraging the motility promoter.
Oh, really?
Yeah, like that was like because it's such an important and robust promoter, right? Like when you do want to swim, you have to turn it on, and it's very highly expressed because you have to make a lot of these flagellin proteins to build a whole flagellum and everything. And so what we did was, instead of having the bacteria express like all this flagellin, we had it express our protein of interest. And so we knew it would make a lot of it, but then of course we had to make sure that it was willing to express this protein and use this promoter even when it wasn't in an environment that the bacteria recognized as suitable for swimming. And so that required me to tap into my understanding of bacterial genetics to know like, how is it normally turn this off, and like how do we sort of convince it to not turn that off anymore so that it will be constitutive, so it'll express it all the time. And so ultimately, fundamentally, that is the core of Zbiotics technology, is that promoter system. And then whatever gene we pop into the slot behind that promoter, right? So naturally, that gene is a flagellin
motility
gene. So, right, exactly. And so now we're just swapping that out for, in the first case, an enzyme that breaks down acetaldehyde. But
so you're not making them motility. So you're not making them extra motile or anything like that. You're not making them swim around in our guts. You're just taking that motile gene and then replacing it with the acetaldehyde enzymes that you got from the probiotics. Or where did you get?
So this one actually so you're exactly right. And so the bacteria actually no longer are motile because we've swapped out their gene for motility, which is fine. It actually makes them, that if anything, it's it's, it weakens them in some way. And so
Oh,
not that it's an issue one way or the other, because this is a very safe bacteria. And so it's not an issue, but like, the last thing you want to do, right, is create something that has some sort of advantage. And so if anything, we've made this sort of have something of a disadvantage.
okay.
But yeah, so in any event, it's no longer able to swim, but it's able to make a lot of acetaldehyde dehydrogenase. And so we get this acetaldehyde dehydrogenase enzyme. This one specifically we got from another bacteria. So what's cool is that like this enzyme class, acetaldehyde dehydrogenases. So these are enzymes that turn acetaldehyde into acetate. They are extremely ubiquitous in nature. Like 70% of all life, not just bacteria, but like all life on the planet has some version of this enzyme. So like
And our livers do too.
Well,
right?
that's say the human liver enzyme. Humans actually have several acetaldehyde dehydrogenases, but like the one that's important for alcohol metabolism, DH2, is actually like not that different than bacterial enzymes that do the same function. And so it's a very old, evolutionarily speaking, a very old enzyme or old gene. And so that's cool. So the things is, they all have these minor differences, in terms of their bases and their charge and some of their structure and stuff that makes them suited to different expression systems that are sort of built. I can think of a good analogy for why this matters, but like they're just like the structure just kind of, it has like the branding of that bug as opposed to like the brand of another, so, I guess you could, like if you get an email from different companies that sort of look differently, but they have the same function for you, which is to keep you,
Informed.
buying their product or whatever. Yeah, exactly.
Yeah. Maybe informed.
Yeah. Right. Yeah. So, basically looked the most like our bacteria, and that our bacteria was kind of willing to express. And so we tried out a few different ones. And the one we found that seemed like our bacteria accepted and liked the best was this one from another just sort of environmental microbe. It's called Cupravatus nicator. And it's, found in like sort of just, water systems and things like that. And it's non-pathogenic or anything, which is important. So, because I actually found a great version of the enzyme in Vibrio cholerae, which is the bacteria that causes cholera. And even though that
Really want that.
it would have nothing, it would have no like, it wouldn't, it's not the thing that makes cholera, dangerous, right? But it's like, just the optics of like having taken it
from a brand perspective. You do not want to be taking the cholera genes from cholera and putting it into humans now. So you found this very like safe version of the acetaldehyde.
Right, exactly. And so we slotted it in there. So we started with GFP, with green fluorescence protein. And so we just showed that we could. And so what's cool is this is just like an awesome protein that like glows green, and the more protein you have, the brighter green it is. And so, when I started building the promoter system, I was just using GFP, and I just kept going until I saw that I got a lot, like a really bright, like green glowing bug. And then we swapped in this.
So that's how you knew you were targeting the right place.
Exactly. Exactly. Yeah. We validated that promoter that way with GFP, because it's like a really easy readout, and it's very dramatic and like, fun. And then we swapped in the acetaldehyde dehydrogenase, and like I said, we tried a few, and then we this one we swapped in and got really good protein expression, and we validated that the enzyme was functional and it could break down acetaldehyde, and indeed it could break down enough acetaldehyde that like it would be relevant, like based on the amount of acetaldehyde you're exposed to when you drink and things like that. So
and it wouldn't decompose in the gut too fast or anything like that. I think that's a big. Sorry, go ahead.
Yeah, totally. No, that was like one of the interesting things that like it was like an early insight that I had when I started building the product was when we started, I really wanted to just keep everything very simple. Like there are a lot of unknowns and challenges and complexities that arise, like when you're talking about the microbiome and all the community of bacteria that live in your gut, and then even when you're talking about bacterial genetics, right? Like there are thousands of genes and they all regulated differently, and so it's like, how could we sort of just pick something that didn't like bring in all of those complications, like that really was like very isolated. And so it was like a single enzyme performing like a single biochemical function, rather than trying to get something that could do a whole bunch of functions strung together, because that's more places where it could break. And then the idea was also like you said, like the environment of the gut can be very sort of unfriendly or like caustic to different enzymes and stuff. And so enzymes break down quickly, the pH is relatively low, and there are a lot of other proteins that break down proteins called proteases. And so you can lose this function quickly if the enzyme is not functional or robust. And so one of the things that I saw was like a cool opportunity was, rather than have the bacteria secrete the protein into your gut and go and try and find acetaldehyde, and then deal with all the kind of like, unknowns and crazy environment of your gut, it would be a lot simpler if we could instead get the bacteria to take up the acetaldehyde, rather than spit the enzyme out. So
Because then it
that
would be in
actually
a
ended up
known environment, so it can
exactly the
process it instead of
bacteria.
having to, yeah.
Exactly. So the bacteria inside the bacterial cell is ideal for the bacterial proteins to function, right? Like they keep the pH normalized and they have cofactors that are available and it's tied to other metabolic pathways. So you get a lot of benefit there as well. And so there's sort of like this wild unknown outside, but then inside everything is like a very controlled little bacterial bioreactor essentially. And so that
Nobody had tried that before.
well, I don't know if anybody tried it explicitly before. It's
I see.
nobody tried a lot of what we've done before. We are the world's first ever genetically engineered probiotic. So nobody's ever engineered a probiotic to perform a specific biological function and then have you eat that bacteria to temporarily gain that function. And so like people have made enzymes using bacteria and then given you the enzyme to eat, right? But that is going to be a much more transient and unpredictable situation. That enzyme
Mhm.
is going to break down in your stomach. It's only going to remain functional for a short period of time. It may not even be functional at all, but like with a live bacteria performing a function for you, it's not just relying on the enzyme. You're getting the whole benefit of the bacteria and all the things that's naturally evolved to do over the last 3 billion.
And so does it seek out the acetaldehyde in your gut, or how does it find it? Because you drink this before or during drinking or maybe after at the end of the night, and then it does it seek out the acetaldehyde that's in your gut?
So ideally, you drink Zbiotics before you start drinking, so it's in there and ready and waiting for when the
Okay.
acetaldehyde is, it becomes available.
Mhm.
And then while it's in your gut, so the volume of your gut is sort of like think of it as like these little like pools of liquid, and and the bacteria kind of like so as your gut moves along, it moves these pools of liquid along, and so the bacteria are kind of like in that pool. And acetaldehyde was a great place to start because acetaldehyde is highly soluble and it can actually passively travel through membranes without active import. And so basically what happens is that the acetaldehyde is constantly normalizing out externally and internally within the concentration of across the membrane of the bacterial cell. So
Mm.
as long as like the bacteria are in that pool with that acetaldehyde for enough time for that concentration to normalize, as they break down the acetaldehyde inside the cell, more acetaldehyde diffuses into the bacteria, and then it continues to break that down. And that may sound unlikely potentially, or like, I don't know, will that really work? And it was a good question. I was worried about that as well. And so when we did our initial testing, we did see that like if we put in a physiologically relevant amount of acetaldehyde, meaning the amount of acetaldehyde we know is probably in your gut after a night of heavy drinking, and we put it in like a known volume of liquid, would we see over a relevant period of time, like say like the 6 to 8 hours that you're sleeping, would we see that acetaldehyde go away?
Mhm.
And indeed we did. So we know that since we have a pretty good idea of what the environment of your gut looks like, we know that when we replicate that it does work. So obviously we haven't gone into a person's gut and like observed
But you
that,
have tested it yourself.
right. And so then the readout becomes different, right? So that's the biochemical readout, like
Mhm.
in the test tube to say like, okay, yes, there is like the data is consistent with the hypothesis that it could help, right? And then, but that is by no means proof, right? And so a lot of people ask me that, like, oh, well, like, yeah, but like, how do you know? And they keep asking like the deeper, but it's like, look, at the end of the
Mhm.
day, like, does the product work for you, right? And so that's one of the reasons why I chose, we have a lot of big visions and dreams for what we're going to do with this technology, but we wanted a proof of concept and a place to start. And I thought a really great place to start was one in which there was a very visceral readout for the end user, right? So like I can
Mhm.
put my data on my website all day, but 95 out of 100 people or 99 out of 100 people aren't going to go look at that data. If they do look at it, they'll never be fully convinced, right? Like
Especially
just
if it's like a normal probiotic that's just supposed to like help your gut function a little bit better, like
yeah.
that you're not going to experience that. But by curing a hangover or preventing a hangover, and you drink a lot and you're like wake up fine, that's very visceral, like you just said.
That's what I mean. Yeah. So the idea is like you can try this product for yourself. And then if you wake up the next day and you feel better, then
Mhm.
like that, all the like biochemistry and the acetaldehyde and like all that whole story, like, I mean, doesn't even matter anymore, right? Like all
Mhm.
it matters is, and if you don't, then okay, then it's not for you. Right. And so that was kind of the idea was, right. Like exactly. If we started with something for general sort of gut wellness or something, there's no way for you to know if the product is actually benefiting you. And people
Mhm.
are comfortable with that to some degree, right? I mean, like there's a whole probiotics industry predicated on just sort of the the belief that these products will help you in some way that you can't know. And like, I wasn't really excited or comfortable with that. At least for the first product, I really wanted people to feel it for themselves. And so that's
So,
one of the reasons why we started there, was like give people that opportunity to really try it and trust their own experience as opposed to just whatever data we put out.
totally. So what was that first time that you tried it like? Did you try it
Yeah.
with a bunch of people, or like did you wake up and what was that like, that moment?
Yeah, it was funny. So I started Zbiotics, and at first it was just like me in a lab for like a year, just trying to
Mhm.
build the product, right? And it took almost to the day, like exactly a year. And so I finally got this prototype, this strain, and I was like, okay, like it kind of like dawned on me, like actually a couple days after I made it, because I was just so in like lab mode and I was like testing. I was like, wait a minute, like I actually have the product, and then like I was like,
Yeah.
I should try it, right? And so like
Yeah.
so it was like a couple of weeks before my birthday. So I was like, okay, I'll try it the night of my birthday. So it'll be a great birthday present. And so I chose to not think about what would happen if I found out that I had wasted a year of my life. So I basically wrote out a protocol for what I knew would make me feel like miserable the next day.
Mm.
And so it's like a drinking protocol, like, okay, this many drinks in this amount of time. And if I do that, there's no way I'll wake up tomorrow and feel okay. Like I'll definitely feel
Mhm.
bad. And so I followed this like protocol, and I have it like I was like writing by hand. And so it's really funny. You can sort of see as like the night goes on that like the handwriting gets like messed up, and like,
As you're like
yeah, it's pretty
tracking
great.
all the drinks that you've taken.
Exactly. It's like simultaneously very scientific and completely not scientific at all. And so yeah, so I did that, and then I woke up the next morning, and I was like, it was great because I felt like I woke up and I felt at first like, oh God, I'm like pretty tired and I don't know, and I kind of got like a mild headache, and I was like, I don't know. And then I just sort of like, whatever. And I kind of, went back to bed for another 20 minutes or so, and then got up and like had a coffee, had some breakfast, and it was like 10:30 in the morning, and I felt perfectly fine. And I was like,
Wow.
oh my God, like that's amazing. And it was great. Like I'm actually in hindsight was very glad that I felt the things I did feel, because that was sort of like a positive control, right? Like we really make a point of saying that this is like, we actually don't refer to it as like a hangover cure. And like it's not a get out of jail free card, right? It doesn't deal with everything you might experience, right? Like this product is specifically engineered to break down acetaldehyde, and acetaldehyde is an important part, but only part of the problem, right? So like alcohol itself, for instance, causes poor sleep. It
Mhm.
also causes like various endocrine imbalances and all sorts of things like that you will experience, like grogginess the next day, like mental fog, like other things, right? And I felt those things. But the good news is those things are relatively easy to deal with, right? Like a good cup of coffee for the grogginess, and a good meal to deal with some of like the blood sugar issues, like the pendulum swing you might have been dealing with the night before, and you'll probably feel a lot better. Whereas like acetaldehyde causes like all these other like really miserable like feelings
Mhm.
of like, like just that toxic kind of death
Yeah, I
feeling and
was
the
looking up the
malaise.
side effects of the acetaldehyde, and it's just terrible for your body. Like that's the
Yeah.
worst part about drinking, and that's why it's the worst part about the hangover for sure.
Totally. Exactly. And so we thought like, okay, well, we can even just deal with that. Like we're doing good. Right. And
Mhm.
so, and that was what I experienced, and it was great. And so then obviously, an N of one is not proof by any stretch, but
Yeah.
at least at that point, again, I always like to say it's like the results were consistent with the hypothesis that the product could be beneficial. And so then, we expanded out from there. Like, had a few of my friends try it, and then they had a good experience, and we just kept, and then by the time we launched, we had done beta testing with, I think, almost 10,000 people, like, really tried to hand them out and get as much feedback as we could from as many people as possible, tons of conversations. So that was valuable, not only to find out people's experience with the product, but just like also their experience with hangovers and like expectations. Like, so that's the thing is like, we don't ever use the word hangover, because in talking to all these people, we found that like that's a very loaded term that has different meanings for everybody. And it's
Mhm.
not a very accurate or precise term. And so, we found that, it was actually a lot better to talk about it in terms of like the things that you could do the next day. That was like
Mm.
much more important to people than like sort of this get out of jail free card. Like this idea that like, you want to go out and have like a responsible night with your friends and, have a few drinks, but then the next day you might wake up and just like not be motivated or not feel like you want to go and do the things you plan to do, like your morning workout, or like go even just meet up with your friends or whatever that. And the key thing that people said is they just hated losing a day. That was like their
Mhm.
big thing, and that's what this product allows you to do. It's not a get out of jail free card. It just allows you to not lose a day. You're not stuck on the couch anymore. And so, so a lot of those insights happen from just a lot of talking to people early on.
How did you reach 10,000 people before you launched?
I mean, partially it was because it took us quite a while to launch the product, even after we had that first prototype, because there's a lot of things that go into it. Like, it was the world's first ever genetically engineered probiotic, which means that we had to figure out our regulatory pathway. We had to figure out a whole new manufacturing system. Like, we're also, as far as we know, like, it's not very interesting and we don't talk about it, but we're the world's first ever shelf stable at room temperature live probiotic beverage, right? Like that, like, yeah.
That
And
doesn't
it's like,
sound cool, but that's huge.
exactly. It's like, it's logistics, right? But like, we wanted people to be able to not have to refrigerate it and have it be stable and have it be stable and pass to your gut without encapsulation. And so all those things took a lot of work. And so, like, it's sort of like blessing in disguise. That took like 2 years for us to do all that. And so in
Wow.
that time, we were just handing out lots of samples. And so we did all kinds of things. We would go to bars and we'd ask if we could set up like a table and like hand out samples to people. And so we would just like pitch. And it's amazing to me. One of the things I thought was like really fascinating was it was amazing how skeptical people were. And I appreciate that very much, because like I realized I would be too if I was like walking to a bar and some guy was like, "Hey, try this thing," and
A
like,
magic liquid. Yeah.
what is
I
that? Like,
genetically engineered this.
oh, it's free. And they're like, okay, even more so now. Like, what is
Yeah.
in that? Like, and it was funny. We learned a lot about like people, like, and so we heard, it was funny. We thought like, oh, we're giving away free samples of like this product that's supposed to help you feel better the next day. And you're about to drink, like, this should be, I mean, the easiest
Easy.
thing in the world, right? Like, it was just obvious. And it was like, there's so much skepticism in the space. There's so much snake oil. And like, people were just and so that was awesome, because we just like learned a ton of stuff about like,
And who is we at this point?
at this point, like at that point is really just me and my co-founder, Steven. And so he's our chief operations officer. It started with just me in a lab as a scientist. And then, we had to turn that I had to turn that science experiment like into a business, right? And I didn't have any qualifications for any of that. And so I needed a partner. And so Steven came on and he helped me, take this to the next level, deal with like all the kind of logistical things that we needed
Yep.
done, like regulations, manufacturing, IP, all those things. And so, but he also didn't have a background in D2C or marketing or branding. And so we were really figuring that out together on the fly. And it just started with just talking to as many people.
Sounds
So
like doing the classic YC do things that don't scale kind of thing. Going out.
Right, 100%. It was exactly that. And it was just like driven to our heads over and over again. That it was okay that this took a lot of time, because like it was so important for us to be getting that direct feedback and that direct interaction. And it did.
And did you tell them to like text you the morning after, or how did
Yeah.
you get the data afterwards?
All different kinds of things. We tried like texting, or like we get their email when we hand them out the sample, and then we'd email them like a survey, just all different kinds of stuff. And it was crazy. Like it was very surprising to me how hard it was to get people to try the product and then to get them to like give us feedback. And so actually though, people were really willing to give us feedback once we convinced them. Like, that's the thing, is like, if I could have a 15-minute conversation with somebody, I could usually convince them like that this was at least worth trying out, and then they'd be like excited or interested. And then they would totally be, now they're part of the science experiment. So they'd like, they would like to give this feedback. But it was interesting to see. And then, now of course, like we don't have 15 minutes with every person. And so it's like, how do we boil down the important parts of that conversation? Like, what were the critical points? And like, how do we get that into like very concise messaging that somebody will see and have that resonate with them?
And how do you scale that on the website without actually meeting these people in person?
Totally. Or like even harder, like in an ad or something, where like you really only have like, a couple of seconds, with somebody. So it's hard, and we're still working on that. But
And so you launched slightly before COVID?
Yeah, we launched in August of 2019. So we had a a good amount of time before COVID, like 5 or 6 months before everything really shut down. And so we're just sort of like getting momentum and getting moving. And like we based everything around special occasions, right? Like you're going to that wedding, or that weekend trip with your friends or whatever, all these like big special moments, right? And then like all of that just stopped existing, right? And so I was like, okay, so that value prop no longer makes sense. But in a lot of ways, it was a good thing. Again, maybe a blessing in disguise. Like what happened during COVID was that everybody started changing behaviors, right? And they were thinking about their social lives differently. They were thinking about how they consume alcohol differently. And so it forced us to really dig in and figure out like, how do people really think about drinking in their lives? And particularly now when they're forming new behaviors, how can Zbiotics it was an opportunity for us to become like a part of that. And especially because, like I say, we're really trying to focus on the positive benefits of the product as opposed to just sort of like pain avoidance. And like that really allowed us to kind of start to kind of dial in on that a little bit. And so it's been a really cool experience.
And so quick question with probiotics, does it stay in your gut? So what if you take one and then maybe the next day you drink again? Are there still some there? Are they replicating in your gut, or how quickly does it kind of go away? It's just like a logistics
Yeah,
question.
logistically, it should about 18 hours on the short end. And that's like
Okay.
normally how the transit time of the bacteria through your gut. We specifically picked a bacteria that is known to not seed your gut, meaning like take up residence there semi-permanently or permanently, because that's actually not something that is easy to control. It's very variable. And so anything that a bacteria does is going to be dependent, at least in part, on the environment of your gut. And everybody's gut is different. And so the existing microbiome and sort of the gut conditions and are all going to be very unique to you. And indeed, like you today is going to be very different than you in even like 3 weeks. And
Mhm.
so trying to find something that would have some level of consistency. And again, this goes back to simplifying, keeping everything as simple as possible. We picked a bug that people naturally eat every day of their lives. It's ubiquitous in nature, and it's naturally evolved to pass through your gut and pass aside and not seed your gut. It's not part of an adult a normal adult microbiome, but it's also not at all dangerous. There's no pathogenicity. It's something you already experience every day of your life, and so that was perfect for me. It's like, right, it's like something we know has a defined and pretty consistent like transient timeline. And so
Mhm.
for some people, it can last a few days. And so, you see, and so some of these careful studies we see, I don't want to mislead. We did not, this is drawing from literature that already existed. So we being generally the scientific community, we see that the bacteria can persist for maybe up to 7 days in some people, but that is very unusual. For most people, it's like 18 to 24 hours.
Okay, so
It's
it
so I
doesn't give
guess
you
like,
permanent superpowers
right.
just for a little bit. Okay.
So for most people, it's a day, right? And then like,
Yeah.
yeah, so theoretically it could last, 2 days for you. And so if you take some, then like, and you drink again the next night, like theoretically it might still be in there, but it's hard to predict at that point. And so we typically just say like, it's about a day for most people. And that
Okay.
is, and there's no harm in having more. So the amount that we're giving you is so because what's interesting is the biological function it's executing and not the number of bacteria, which is like,
Hmm.
you see all these probiotics and it's like a billion, 10 billion, like 100 billion. And you're like,
Yeah.
do I need that much? It's like, I don't know. And so like our dose is much smaller than, or it's not a dose, like our CFU count is much smaller than like a traditional probiotic because it's the biological function that matters. And so, we tested it like 1,000-fold higher CFUs than what we have in our product. And, with no safety issue whatsoever. And it's indeed consistent with like naturally fermented foods like natto and kombucha that have this bacteria already in it. Well, not our
Hmm.
specific strain, but like this species. And like, we're even lower than what you would just normally encounter in food. So having
Nice.
multiple was not a problem, but you don't need it.
So what was scaling up the manufacturing process like?
Yeah, that has been like probably one of the biggest headaches for Zbiotics, honestly, because we have this live probiotic bacteria that's genetically engineered and it's so stable. So this bacteria we use is called B. subtilis, and it's this common ubiquitous environmental microbe, and it has this amazing capability of being able to form a spore, which allows it to be really resistant. So they've pulled like this bacteria out of like ice flows from like 10,000 years ago, and they're still alive. Like they can
Oh, wow.
last forever, and they're really stable in that spore form, which is great because it means when you eat it, it already comes with its own encapsulation. So it is naturally resistant to the acid of your stomach, and then it reaches your gut and it like wakes up. It comes out of that spore, and then it starts functioning in your gut. And this is like just such a
And so that's
cool.
also how it has such a stable shelf life too.
Right, exactly. So we didn't have to build that, and nature built that for us, which was great. But what we had to do is make sure that we preserved that natural function when we were designing our manufacturing process. And so that
Mhm.
required us to because we wanted to bottle it in a beverage and we want it to not have to be refrigerated, we have to make sure that there's no other like contaminating microbes in there, which is like goes without saying, obviously. But the way you normally do that is through pasteurization, which is like killing all the bacteria. We
Mm.
didn't want to kill our bacteria. So we had to like figure out a way where we can make sure that we get no other bacteria and then we killed all the other bacteria, but not ours. And so
Mm.
that was like a big challenge in and of itself. And then we had to find somebody who's willing to work with this very resistant spore that theoretically could cross-contaminate other products they had or whatever, because we don't want anything else in our product, but other products don't want our bacteria in their thing either. So we had to figure out a way so we had to find somebody, first of all, who's willing to work with us on this. And then we had to kind of develop a new pathway. And so we found somebody, but they're very small scale. And that was okay to start, but it means that, we're constantly trying to figure out ways to scale up the process and like grow it. And so it's been a big challenge. It was we have these very narrow guardrails we have for our product and these requirements. And it ruled out the whole world of like contract fermentation for us, except for just a very short list of providers, which was
Did
hard.
you have to make your own manufacturing, like new manufacturing processes for this, or were you mostly able to contract it? And then I think you're also looking to like vertically integrate as well.
So eventually I think one of the things I learned from this, and I never thought this would be the hard part, was the manufacturer. I was like, we've been growing bacteria and fermenters for like ever, like, I was
Yeah.
like, like brewing beer is just like, I mean, it's yeast, but it's like same thing, right? And so, and it was amazing how hard it was to find. So yeah, so right now we've contracted it out and we developed a proprietary process with contractors, and we do have providers, and we're trying to build in some level of redundancy, because what happens is we'll find somebody who can do it for a while, and then for one reason or another, they won't be able to do it anymore. Like we have one contractor, they were based in Canada. They were actually like a subcontractor of the Canadian government. And then we paid all this money to like get the process up and running. And then like a month before we were ready to launch, and they were just about to doing our first full food grade run.
Mm-hmm.
They just like called us up and they were like, hey, the Canadian government decided that this facility is not really, viable, so they're shutting it down. Right?
Wow.
And like, so we're like a month over launch and we have nobody to manufacture, even though we've done all this stuff with them up until that point, right? Like to get ready, and then it's just all out the window. So then we scramble, we find somebody else, luckily, and we had somebody else. We were already looking for a backup, not that we even had any inkling or idea that they weren't going to be able to, but we wanted to have redundancy. And so so luckily we already had this other company on the, in process, and they were like willing to get up and going really quickly, but they kind of put us in one of their pilot facilities, small scale, because that was what they had available. And then COVID hit, and we, reached out to them. We're like, hey, let's do another run. They're like, we switched all of our equipment over to COVID manufacturing. So they were doing different drug development projects and vaccine development projects with other companies because they're biotech. And we're like, great, thank you for doing that, but also darn, because like we need to find a new partner. So we, so it just keep kind of happening like that. Is that like, we find these people and they're sort of or these companies, and they're using sort of like atypical parts of their process and slotting us in. And so
trying to build your own process is the goal,
ultimately,
right?
To avoid all that headache. Exactly. We want to have our own line, like, because we have a unique fermentation process and we have a unique bottling process and all of that like it just makes sense for us to bring this in house. And it's also very expensive though, and difficult to do. So we're not ready yet, but we've looked into it, and that's the thing we're starting to kind of figure out. And so in the meantime,
nice, for sure,
contracting. Yeah.
Was that the point at which you were sleeping on the floor of the lab, or was that the point when you were developing?
It was that first year when I was developing the product. Yeah. So it was just me in a lab, and I had nothing else going on. So when I started the company, I had another job, and I was in Miami. And then I applied to Y Combinator's fellowship program, which was just basically a $20,000 grant to just sort of like to help companies get from 0 to 1. And I got that. And then I just, after about 12 hours of soul searching, was like, no, yeah, I'm doing this. And job. And then like started Zbiotics, but I didn't have a lab. I didn't have anything. And so I managed to like get a lab out in the city of Berkeley. And, and I'm originally from Northern California. And so I like just flew home or drove home and from Miami, and like stay with my parents and in Sacramento. And that's about an hour and a half from Berkeley. And so I was like, because I didn't have any money to pay for an apartment or anything like that, because $20,000 is not.
No, that's not enough for rent.
Yeah, exactly.
No, you want to spend it on the lab, right?
On the lab. Yeah, it was all 100% going into R&D, right? So what I would do is I would just like drive to Berkeley, and then throughout the week I would, at night, like I say, I had nothing else going on. So I would just like roll out the sleeping bag under my lab bench. And I don't think that the guy who was running me the lab space knew I was doing this. And so, but I mean, it's fine now, right? It's over. But like,
that's awesome.
I wake up at like 7 a.m. and, before anyone else got in, and like clean up and then just get back to work. And it was a really fun time, because the problems were problems I understood, right? It was science. It was things that like I could just work hard and figure out. And then, ironically, now that I work a more, normal hours and I don't sleep in the lab or anything, I feel like the problems are a lot harder, so I'm like kind of, and there's so many things that we had, so many more problems we have now. And many of them are not in my area of training or particular expertise. So, yeah.
So now I guess your main focus, now that you have this first Zbiotics product, is that you're working on new product. So what like future product lines for, I on your website, it says you're looking to enhance humans, but then also to create new materials and to sequester carbon. Like let's start talking about the enhancing humans ones. What else can we do other than preventing hangover or reducing the acetaldehyde, not preventing hangovers, but almost preventing?
Right. Yeah, totally. I mean, I think that's, that is ultimately what, right, we started the company for, right? This is always meant to just be a proof of concept, right? Like a first way to introduce the tech, but is the world's first ever genetically engineered probiotic, but it definitely won't be the last by us
Hmm.
or others. And so I think this is just, as I said earlier, it's like we can program any biological function on the planet into a bacteria, and then you can eat that bacteria and temporarily gain that function. It's like, what a powerful idea. And
Hmm.
so we do have lots of ideas of things we want to do. Like our second product, we're in the process of commercializing. And so I can't say exactly what it is yet because we're still trying to keep that powder dry, but it's not at all related to drinking. It's much more related to the things you might expect. Like it's related to like gut health and the microbiome. And so really tapping into my microbiology roots and making a better product than the other things that are out there. And so generally, like within that realm, we're also developing products that are related to better nutrient absorption from your foods or protection of normal toxins you're exposed to every day, which is a lot like the acetaldehyde product. But like, you think about like heavy metals in your drinking water or things like that, things that you aren't able to really avoid, but that we also
Hmm.
aren't good for you. And like, right, we have microbes deal with this stuff all day, every day. And so like, we can leverage that. And so those are some of the things. But then I think what's even cooler is that like we also know and are continuing to learn more about how the microbes in your gut really influence so much more about your health in your body than just the gut. And people make too big of a deal out of that or kind of like overclaim there. And so, and
Hmm.
that is frustrating. And so I don't want to like, maybe like, try and sell the world here or anything, but we do know that, for instance, like we know there's a good literature that shows that like, certain byproducts of bacterial metabolism help your body recover better from stress related to like exercise or poor sleep or things like that. And there's good documented literature to demonstrate that. And so we can leverage and enhance that. So we can help you with engineered microbes recover better from different stresses you put on your body through normal living, through either exercise or performance enhancement, or
So like, can that help
through
your
sleep deprivation.
so can that kind of help your muscles grow? Is that like if you're
Like
recover from exercise, or yeah, go ahead.
exactly. Yeah. Exactly. I mean, so like it wouldn't be, I mean, I don't know, who knows, right? Like there's a biological function of everything. So maybe help your muscles grow, but like, even just like reduce the things that hinder your muscles from growing, like if that makes sense, is like a fine nuance, right? Like
Hmm.
if there are byproducts of your workout that strain your body or hinder your body's ability to recover, there are bacteria that can help you deal with those byproducts, those stress related byproducts. And so that can at least just like clear the way for your body to heal itself more efficiently or better, for instance,
Hmm.
after workout, after poor sleep. So it is really amazing. And then we also know, like, there's a lot has been made of like sort of the gut brain axis, and a lot of the data is very preliminary, but there are things that we do see in the literature related to mood that are direct biological outputs of, or metabolic outputs, I should say, of the bacteria that live in your gut. I mean, so
Hmm.
there are things that we could potentially do related to mood. And then I think one of the things that I'm really excited about too is like, and this is like really far afield, right? But like the idea of
That's
like,
awesome.
astronauts, right? And like
Hmm.
someday we're hopefully going to go like semi-permanently into outer space, like Mars or whatever that might be. And there's a whole new range of, right, stresses that you're going to have to deal with at that point, right?
Hmm.
Related to like radiation exposure, and you're going to have a limited diet, and like the nutrition you extract from food is going to be so critical at that point, right? Because you can't just go to the grocery store and get a convenient bag of like vegetables, mixed
Hmm.
vegetables and stuff. So like thinking of bacteria that can really help you recycle waste more efficiently, absorb nutrients more efficiently, protect you from radiation. I mean, bacteria have all these amazing genes related to protecting their genomes and exposure to UV, all these things, and that we could theoretically leverage. And so I think
Do
that
you
there's
think that would
just like
also
so
be
much excitement.
delivered? Sorry, do you think that would
No,
also
yeah.
be delivered by a drink as well, like to protect us from radiation, and then it might take the food prep? How exactly would that? That's
I
pretty
mean,
cool.
there's all different kinds of ways, right? Like the bacteria just have to go in a way that they'll have access. So if it's useful for it to be in your gut, and sometimes it is, and surprisingly sometimes it is, that
Mhm.
the gut is actually the source of need. But sometimes you have a microbiome on your skin, and so you can imagine a engineered microbe that you smear on like a sunscreen,
Mhm.
or like you have bacteria like in so many parts of your body, in your mouth, for instance, like they're all, and in your sinuses,
You're
like
gonna
they're.
Zbiotics toothpaste coming soon.
I mean, totally, right? Like we could, right? And like,
Yeah.
cavities are created by bacteria, right?
Mhm.
And so, and bacteria, microbes in general. And so like bacteria can also be the solution to those problems. And so I
Mhm.
think there are just like so many really cool things that we can do anywhere where a bacteria has access, that ends up being the source of a problem. So what we're really doing here is in situ delivery of an important biological function, right? That you
Hmm.
didn't have in that site before. And so that's the power of having a living microbe executing a function as opposed to just like delivering a small molecule or an enzyme itself. Is
Hmm.
that like you have the factory that's performing that function for you in that spot, which I think is just amazing. So there's just so many cool things you
Yeah,
can do.
yeah, and especially if you can make them grow in the outer space, then you don't have to keep sending them up to the astronauts because they'll just keep replicating as well, and then they can maybe keep eating that, and it replicates by using waste products from the astronauts or something like that, and then just converting it
Totally.
into valuable materials and growing from there.
I mean, it's
That's
totally
really cool.
possible, right? Like right now, we focus specifically on transient microbes, but
Hmm.
theoretically, if we wanted to, and I think that there are many challenges that come with this, but like if we wanted to try to engineer something that was a more like semi-permanent resident of the microbiome, like in
Hmm.
the case of an astronaut where that might be really important, then that is a possibility because right, you do have microbes that take up more long-term residence in your body, and so that is a possibility with the technology for sure.
I bet another one would be maybe increasing bone density of an astronaut.
It
Yeah,
can, yeah,
yeah,
it's true. Zero G can be an issue, right? Like in bone density and muscle atrophy and things like that. And so I'd have to think about how, like, a lot of things I've said already are things I've already thought of. I haven't thought of that one. So I'd have to think about how we could target that biologically in a way that was also like safe. But I mean, certainly theoretically it is possible. Like I have to think
hmm
more about it. But yeah, I totally. I mean, that is a major issue. So yeah. So
and
I'm always
and
really
and
excited about the astronaut stuff. I think I
yeah,
wish I
really
could have been
cool.
an astronaut. So
Maybe you can be. Maybe in 10
yeah,
years you'll be able to take a quick trip to the moon or something like that
totally,
pretty
totally.
easily on Starship. What about creating new materials? How can you use these bacteria to create, or probiotics to create materials?
So like, I think when we think of materials, particularly when it says like on our website, is we talk about carbon sequestration and material. And a lot of this is like us sort of advocating for the broader field of genetic engineering. And so one of the things that is really front and center for me when I started this company was that it was very important that we dealt directly with consumers and we had an open and honest and transparent conversation about our use of genetic engineering and GMOs.
Mhm.
And, in a time when, as far as I know, there's no brands or maybe, a small handful of brands that say anything about having genetic, GMOs, where every brand is saying non-GMO, even if it's like water or, some random product that like could never have been genetically engineered,
Yeah, nobody's
non-GMO.
bragging about it.
Right. And we're standing up and we say proudly GMO on it's printed on every bottle of our product, I mean, every box of our product. And it's right in front center on the website. Right. And it's not so much like advocating for the technology, but it's like, let's stop pretending if we're using it, pretending like we're not using it as if there's something to hide. And then if
Mhm.
we're not using it, bragging about not using it, that really conveys a message that this technology is a problem. When in reality, the technology is just a technology like any other. It can be used irresponsibly or for good or bad, right? And so really what we should be evaluating is the end products that are being built with this technology. In the same way that metallurgy is just a technology we use to make different products, right? And then you make a knife or a spoon and they have very different safety profiles and very different things that need to be considered, right? But the fact that a spoon was made with the same technology as a knife does not inherently make the spoon as dangerous as a knife, right? And so I feel like it's the same thing with genetic engineering. And the more people who use this technology hide it, the more people are apt to fear it and reject it, like outright. And
Mhm.
so when we say proudly GMO, it's not to say that all GMOs are good or safe because that's ridiculous, right? Like every product has its own safety profile. And so what we say is that the technology is not inherently bad. And
Mhm.
we try to advocate for that. And I think there are so many important and useful things that technology can be used for. So like with carbon sequestration or with like higher yield per acre crops and drought resistant crops and things, we're protecting humanity from starvation, right? Like as
Yeah.
humanity grows, like that is a very important thing that needs to be done in one way or another, right? And going back in time to organic farming, and this is not me saying that organic farming is bad. There are a lot of important things and techniques that can be leveraged from organic farming, but going back in time to 17th century technology to feed like a 21st century population is not the answer. It's also arrogant and not inclusive, right? Like, oh, how nice it is for you to be able to pay extra for your food. But the fact that you pay extra means that it's not sustainable, right? Like if it was sustainable, it'd be cheaper. It's more expensive because it's less efficient and less sustainable, and does not provide democratic access to resources. So like we do need technology to improve, right? And so it's just a matter of how that improves. And so we believe that if we are able to elevate this conversation from is it or isn't it, and like good or bad to something that's like, okay, this technology exists and how do we use it responsibly and transparently in a way that people are comfortable with that also can help benefit humanity, like with things like new novel biomaterials replacing chemical materials, or just slaughtering a bunch of cows to get leather. Why don't we use engineer microbes to do that? Like that's a way more efficient and humane way to get that thing done. Then we believe that like that's part of our mission, is just really elevating that conversation for the technology as a whole.
When do you think you'll start making like a carbon sequestration microbes? Do you have a
I
timeline on
mean,
that?
like we are not like currently that is not like
You're
in
just
our
advocating for that in
pipeline,
general.
and we're really focused on exactly. And like we know that there was this amazing paper that was published where they made E. coli get all of its carbon from sequestering it from the air, as opposed to from sugar that was put into the media. And so like, that was amazing, right? It was able to completely function by carbon sequestration. And so as that technology continues to advance, that is something that we could potentially start to leverage when it comes to dealing with the excess carbon that we are currently releasing from underground back into the air, right? That's where it was before too, right? It was in the air, and then a bunch of dinosaur bones or whatever, like you sequestered it as oil, and now we're releasing it back out into the air by burning it. And so we're like, oh, we probably shouldn't. We need to put that back underground. And so finding biological ways to do that through genetic engineering, I think, are possible. And so yeah, exactly. So I think our role within carbon sequestration, I see it is really just like advocating for the safe use of the technology and regulation. Like that's the other thing, is we really want to define clear regulations around, because all it takes is one bad actor, right, to use this to engineer a microbe in a way that ends up being unsafe or a disaster, for it to ruin it for everybody. And so it's actually really important for us to be responsible, and we also see like the need to advocate for clear, logical, and science-based regulations around genetic engineering and genetically engineered probiotic microbes in particular. And so when companies are out there building microbes that are engineered to sequester carbon, they're doing that clear expectations about what they should be looking for in terms of safety and responsibility. And so I think that totally can be done, of course. And so like that's what we want people to do. And so we're in a conversation with a lot of folks about how we accomplish that, like companies and the government and things like that. So
Nice,
really excited
nice.
about it. Yeah.
Well, let me know if you end up doing anything with the Salton Sea. The Salton Sea in Southern California, it needs to be terraformed, and I'm very obsessed with learning how to terraform this. And essentially, it was a man-made sea in around like 1905. The Colorado River burst and then flooded the Salton Basin, and it created the sea. And for up until the 1950s, it created a bunch of life and biodiversity flourished. But because there was no inlet, the water kept evaporating, and then eventually the salinity of the water, now it's almost twice as much as the Pacific Ocean. And so there's toxic algae blooms, there's high salinity, nothing can grow there. There's dust pollution, so asthma is like 3 times the rate in the surrounding areas. So I think that because it's super high salinity, maybe you can have microbes that can deal with maybe decreasing the salinity of the water, or fixing plants near the barren desert or something like that. I don't know the exact solutions, but I am very interested in terraforming the Salton Sea. So maybe when I have a ton of money, I can order some microbes from you to work on this.
This is like so fascinating. Like, so like one of the things I thought was related to is like, I went to the Dead Sea, which is, a very high salinity sea. And like, they thought for a long time that there was no life there because it was like so salty that nothing could live. But then of course, like everywhere, they find that like there are microbes that can live there. And so, and which is amazing. It's like, how do they do that? And like, they have these crazy osmotic like pressure gauges and things like that. And so, it makes sense that we could pull from these other environments and these other biological functions to make something that theoretically could, like you say, maybe like, it's funny to call it like terraforming, but like, because it is on Earth.
Terraforming is totally what it is, because we created that in the beginning, right? In the 1900s or 1900, it was fine, and then we created that, and then we let it get worse, and now we have to, now that it's there, we're like, oh crap, we should make it better. Like we shouldn't just watch it.
Totally.
I think humans have the tendency to just watch things get worse and be scared to make things better. In terms of like in this kind, it's like, oops, we accidentally created that. We're going to watch it get worse, but we're not really going to do much to actually make it better. But we need to change that mindset. And so by just talking about terraforming, I think that's super important. It demonstrates agency over our environment, and like how humans are really cool and can create this cool stuff. Totally. I think that's important. Yeah. So when you start terraforming, definitely that's why I'm like, asking a bunch of questions about that because
Oh,
I want
I'd
to
love
order
to.
microbes from you too when you do
Totally.
that.
Well, it could be like
when you
a
can.
kind of thing that like we have all these like great ideas of things we want to build, like ridiculous stuff, like things we didn't even get to this. We just talked about human augmentation, but like, stuff you could like imagine spraying microbes in your shower to like help prevent
Mm.
like fungal growth, right? As opposed to like bleach or like these toxic compounds, or toxic, like harsh chemicals or whatever, and there's so many cool things we could do with microbes, like even outside of our bodies, right? That would be like really, really cool. So, very excited about all the stuff we can build. And so I don't see this as something that Zbiotics won't do.
For
So
sure.
hopefully we get to get into like
We're definitely next time
terraforming.
you're on the podcast, we'll have more ideas about how to do this.
All right, that sounds good. Yeah.
Yeah. So kind of in closing, you started out in classics. If you, I guess we're going over it. Is that all right?
Yeah.
I just
Yeah. No,
wanted
no
to
problem.
just quickly like talk about how you started off in like a classics degree, and then you went into a PhD in microbiology and starts like how do you think that helped you, and how did you get on that path?
Yeah. I mean,
And what
like
advice do you have for anybody else kind of thinking about getting into startups from the research side?
totally. Yeah. I mean, so it's not totally fair to say that I started in classics because I actually did double major in college, like in classics, but then also in molecular and cell biology. And so I did do them both. And I think that they sound very disparate, but, and like recently kind of thinking through how I got there, I think that both of them search to uncover truth from like the unknown. Because like, what I really liked in classics was like, the archaeology. Art and archaeology was my focus. And so like in archaeology, you're digging around in the dirt and trying to like reconstruct, like discover information that will allow you to kind of construct a story or a truth about what happened. And I think that like with science, it's the same thing. You're sort of like digging around in a microscope, trying to uncover information that will allow you to construct what has happened or what is currently happening in the world. And so I went on an archaeological dig actually while I was in college. And I think I saw that it was the way that the stories were created and like the truth was uncovered was maybe didn't speak to me quite as much as the way scientific stories were created and discovered, although inherently very interesting. And I still have a deep passion for classical history. Science was I could tell was my true calling or whatever. And so
I
one
see.
was sort of like an interest or a hobby, and the other was so I spent several years trying to figure that out before I went back and got my PhD. It took me 4 years really to make the decision to go back to grad school because I didn't know what I wanted to do, and I just tried a lot of different stuff, and then I realized that like science was where I wanted to be, and I became very interested in microbes. And initially from a disease perspective, I was really interested in kind of like how microbes cause disease. But then as I started working in biology, my PhD, I realized like, even though I was studying a disease causing microbe, I started to appreciate all the amazing, like disease is actually a very narrow part of what microbes do. Like most of what microbes do is like either neutral or beneficial to humans. And so like, and that's actually a far more interesting question for me. And so I think what drove me there. And then, like, I think my advice was the best piece of advice I could give in terms of like anybody thinking about starting a startup and or going to grad school, like, because you get this question a lot. It's sort of like, should I go? Do I need to? Whatever. It's like, don't go to grad school because it's the next step, or like you think it'll look impressive, or you have to, or something. It's like only go if you really want to, and that the specifically whatever it is you want to do, having that degree will help you accomplish that goal specifically. Otherwise, it's a lot of time and effort that that you may end up not even needing.
And so when you went back to grad school, you knew that you wanted to start a startup in microbiology. You just didn't know
Well,
exactly what it was, or
100% sure that at that point that I wanted to start a startup, but I knew I wanted to work in small biotech of some variety. Like, and I did see a possible manifestation that being that start a startup someday, but it wasn't like I'm going to start Zbiotics, I'm going to get a PhD so I can start Zbiotics. It was just, I was like, I knew that I didn't want to be in academia, but, and also I saw that like big pharma I wouldn't have as much control over my research or like how I applied my science. And so like the idea of having like what I really wanted to do is have a direct impact on people's lives, like myself with my science. And so like I thought that the best way to do that was to make a product or work on something that people would use. And I thought that small biotech and startups was probably the most direct line I could find for that. And that's, that is what I'm doing today, right? Literally the thing I built in the lab, sleeping on the floor of the lab, is now people are buying it, and using it all over the country in their lives. And that to me is like the most thrilling and exciting part about it all. That was it. And so then when I decided to start a startup, again, it was not something that I thought was something I would do someday. And then I ended up starting Zbiotics because like, I just couldn't not start it anymore. Like I kept this idea kept swimming around in my head, and I couldn't shake it. And then my friends made me apply to this Y Combinator fellowship program, and I got it. And it was just sort of, like, I don't know if
you'll end up in a better place if you take that for sure. Like even if it
right.
doesn't work out, like, and you have to see it happen. You have to at
Exactly.
least see how it will happen.
People always say like, oh, just get started with a startup. And it's like, I don't think that that's too simplified, right? At some point, you have to care so deeply about what you're doing and be so excited about it that you just get sucked in by the gravity of it more than, actively starting it. And so I think that that's what happened for me. And that's my advice is if you're thinking about starting it, ask yourself, could you imagine not starting it? Is that even a possibility for you? And if the answer to that is yes, then give it more time, like think through it more, right? But if at some point you're like, no, there's no reality I can see myself living in where I wouldn't start this, then like, okay, then it's probably time to start it.
Did you have other startup ideas?
Not really. I mean, probably. I used to carry around this little book in my back pocket, like one of those little notebooks, and I write like down. It was like titled like million dollar ideas.
I got a book.
Yeah, exactly. And so and I always like find all these like weird little documents like buried in my old computers of like little like just like streams of consciousness of ideas. So like I had ideas for stuff, but none of them were really fleshed out enough to be a startup. I think Zbiotics was the first time where the idea had matured enough that I was like, I want to make it started out sort of like living microbes in your body, preventing disease or doing something for you. And then it was like, what about probiotics you could eat that you are engineered? And so, and then and then it got to like acetaldehyde and that kind of stuff. So like it happened over, honestly, like that idea ended up maturing over the course of like almost 10 years before it like really became Zbiotics. And so,
wow,
I definitely had other ideas.
It needed some fermentation time.
Totally. Exactly the way to put it. And like, I was like, and I never really considered myself an entrepreneur. And so I just like never kept thinking, like, I was just thinking like, this is a good idea. Like somebody should do this, or I should maybe like think of a way to do this. But then it was just like, starting a company was sort of like took a lot of activation energy for me to kind of get to.
Awesome. What's like other biotech areas that interest you other than microbiology? This is the last question. I'm just curious, like where you would be exploring or what else interests you outside of microbiology.
I think like microbiology is probably like, the thing is that within like microbiology or synthetic biology, maybe if I can like cheating a little bit, like synthetic biology more broadly, it's like, it's not always microbes, is so exciting because all the things that we can do, like, right. Like, so I work in like direct to consumer probiotic things foods essentially. Right. But like you can, like we were talking about, like you can have so much of an impact on the environment or like on agriculture or materials. Like, I mean, there's just so many things you can do. And I think that to me is just like,
there's so much in here.
Yeah. It's like, I can't imagine ever leaving because the pool is already so big of things we could do. But, I guess within synthetic biology, there's a lot of other really cool things. Don't necessarily directly deal with microbes, like, non-cellular systems where you make bioreactors with just the enzymes that you want and don't mess with all the other biological functions that a cell does. I think that's like a fascinating thing.
That is really cool. I'll have to look that up.
Yeah.
Look into that.
And then, and then I sit on the board of this company that's like looking for like how we define complexity of life in ways other than genetics. And I think that's like fascinating, so I think within like the realm of greater synthetic biology, there's, now like the bounds are limitless, and it's just like, I mean, it's amazing. So.
That's awesome. Awesome. Well, thank you, Zach. I really appreciate the time. And I look forward to having you back on when we can talk more about some terraforming or other Zbiotic projects. I'm super excited. Definitely go, if you're listening to this, definitely go test out the Zbiotics product and use code NASJAQ and let me know how it is. Definitely tweet at us and give us some feedback. So looking forward.
Totally. Thanks, Jack. So much fun. Appreciate it.
Thanks for coming on, man. I'll talk to you soon.
See ya.
Awesome. So I hope you enjoyed the podcast. That was super fun. I really love this startup. I think they're going to do amazing things. And it's really cool that they're starting with a consumer product and then bootstrapping from there. So remember, go to zbiotics.com, use code NASJAQ for 10% off, and let me know, tweet at me how the hangover felt the next day, how the hangover didn't feel the next day, really. Or just tweet at me what you thought of the podcast, NASJAQ __, and I'll see you guys in the next one. Peace.